Okay, class. So here we go on part two. So in part two, we're going to talk a little bit about ion channels and nerve cells. We're going to start off just with some basics on ion channels. So normally ion channels, these allow free flow of ions across the lipid bilayer. And normally then they're in their sort of closed position. But then they can open up and allow a free flow of ions through. Now, what's interesting is most of them have what's called a selectivity filter. And we'll look at this closely in the next few slides. But typically, a specific ion channel will only allow the flow of one ion. Okay, so if you sort of think about it and think about the periodic table, if you compare potassium and sodium, potassium is actually a much bigger ion. And so you would think that the pore that's created here for potassium to go through, sodium could just flow through as well. But that actually doesn't happen because there's a selectivity filter that prevents only the right ion for passing through the ion channels. Some of the things about the ion channels, typically on the outside where it's sort of adjusted to the lipid bilayer, it's very hydrophobic, and the pore region here that allows ions to travel through is hydrophilic. And once again, the selectivity filter here will determine what ion is actually being allowed to pass through. Now, the different ion channels can be gated in different ways, and what I mean by gated, I mean opened up. Okay, so you can have a voltage-gated ion channel, which when the membrane becomes depolarized, that can open up. You can also have a ligand-gated one where you can get an extracellular ligand to cause the channel to open. You can also have intercellular ligands binding to allow it to open. And then you can have a mechanically gated one. And also, not shown here, phosphorylation can also help regulate some of the gating that occurs in these. And so once again, the gating is just the opening up. So membrane potential becomes very important here. So if you have sort of the exact balance of charges on each side, the membrane potential is 0. However, if you have a net positive charge on the outside and net negative charge on the inside, gives you a sort of a non-zero membrane potential. And the transporters that we talked about in the last section it can help establish these sort of membrane potentials. And if you ever wanted to calculate the resting membrane potential, you use the Nernst equation. I'm not going to hold you guys to that and go over it, but you should have it. And if you're going to end up in a neuroscience lab, you should really know it. Okay. Okay, so the selectivity of ions is a major component of ion channels, and this is actually the bacterial potassium channel. Okay, and so this is sort of a rough schematic of how it looks. And this is kind of important because what you have is a vestibule here where the ions can sort of sit. Okay, and you have these pore helixes here that have this dipole which will actually select for positively charged ions. And then you have this selectivity filter here that will only allow potassium and not sodium or calcium or some other positively charged ion from passing through. And the way it does it is these carbonyl oxygens here can actually select for them, and they're positioned perfectly. So just the pore helix and the dipole, you can actually have this set up by the way the helix is running. So it's got a net negative charge here and a net positive charge here, and this will help select for cations. In the selectivity channel, what ends up happening is you'll have in the vestibule your potassium ion, let's say, associated with water molecules. Okay, now to get through the pore, the selectivity has all of these carbonyl oxygens, so they're perfectly spaced so that they can bind to the potassium. In the case with sodium, even though it's smaller and you think that it could just readily pass through, it doesn't, because in the vestibule, the sodium is also bound by water. And when it enters into the selectivity filter, it can't make attachments to all of the carbonyl oxygens, and therefore it can't pass, and it sort of gets stuck here, and so only the potassium can go through. So that's the selectivity filter. The opposite probably happens with a sodium channel, in that the carbonyl oxygens are actually positioned much closer, and that actually prevents the potassium, which is bigger, from passing through. So this is sort of an important concept that I want you guys to all sort of really appreciate here on how selectivity works, because in general, you know, the potassium channels and the sodium channels, they're very specific on what they can select, also the calcium channels. And so appreciating this really goes a long way into you guys being able to appreciate how the cell selects for it, right? So this is sort of unfavored, and it's going to go back to its water conformation more readily than the potassium, and the potassium ion can just flow through. If we go back a couple slides, you can see how they're positioned here just perfectly for the potassium ion to pass. All right, so once again, you have the vestibule, and then you have your pore helices that are going to select for positive ions based on their dipole, and then the selectivity filter will only allow certain ions to pass through. So in the conditions where they're gated, they can either stay sort of closed, or when they open up, they become open, and this creates sort of the carbonyl oxygens positioned such that a potassium can go through. Okay, so in general, channels can be either closed or they can be open. Here's also how some other ones work. The chloride channel. Once again, there's a selectivity filter that's going to select for chloride, and that's it. And then you have, in this case, your pore helices selecting, so the dipole is selecting a negatively charged ion. And in the case with the potassium channel, it's a little bit different, where you have the negative charge and then your selectivity filter, right? And if you sort of compare this to what we just saw here, it's basically in the same type of conformation. Now, there's also channels that allow water to flow through, and these are called aquaporins. And this is somewhat of a complex concept, but I hope you guys can all sort of appreciate it. So normally, if you just have a whole bunch of water molecules, what happens is protons can jump around, right? You can basically have a bunch of protons and a bunch of OH minuses in solution, and the protons can readily jump. Now, in the case with aquaporins, though, they don't allow protons to sort of move through the channel. They only allow intact water molecules, H2Os. And the way they do it is these asparagines here are positioned with the oxygen on H2O, and that actually prevents protons from jumping. And so this will allow water to pass, but not individual protons to sort of jump through. And so the aquaporins are permeable to water, but not to protons. It's sort of an interesting concept. Okay, now on to some nerve cells. So here's a nice schematic of a neuron. We've sort of seen this before. You have your cell body, and off the cell body you have your dendrites, and then you have your axon. And then if you go down the axon, you have your terminal branches here, okay, and these will join with to help form a synapse. Now, what's interesting, and we'll see a really great slide in a second, these cell bodies here, the dendrites, they can actually receive hundreds of thousands of inputs. But not every input that a neuron is receiving actually results in an action potential traveling down the axon. They're sort of integrated, and we'll see how that happens in a second. We're also going to look at how action potential travel down a neuron. Okay, So ultimately, though, the neurons, they actually receive inputs, and then depending on it, they sort of integrate those inputs, and they'll either fire or not fire, and then an action potential can come down, and then you'll have sort of fusion of neurotransmitter vesicles in the terminal branches of the axon. Okay, so as an action potential is traveling down, it only goes in one direction. You would think it could probably go in two, but it doesn't, and it's part of the mechanism of the channels ensure that it only goes in one direction. Okay, so for example, a sodium channel is only going to exist in three states. Okay, it can exist in a closed state, it can exist in an open state, and then it exists in an inactivated state, and it's in this inactivated state for about a millisecond, okay, or so. And so if you give some sort of stimulating current, the closed channel will then open, ions can flow in, the membrane will get depolarized, and then the channel is actually then in an inactive state. It can't reopen and ions can't pass. Okay, and then as time goes by and the membrane gets repolarized, it goes back to its sort of closed state, and then it can receive another signal. And it's this mechanism here, and this is a very important mechanism that basically allows the action potential to go in one direction and only one direction, and we'll see that in a second. And that's sort of the propagation of the wave. Okay, so the action potential just travels down the axon in one direction because it will become depolarized here, and you would think it could become depolarized over here, but it doesn't. It actually goes in just one direction due to that sort of inactive state of the sodium channel. And then it'll move down here where it becomes open, becomes depolarized, and then it's inactive. And then it can come down here to and depolarize this region. And so to sort of give you a better idea about that, you have your sort of behind the open state where the depolarization, you have inactivated channels. These cannot reopen again. And so as the membrane gets depolarized here, only the ones ahead of it where they're voltage-gated sodium channels can actually open up. Okay, so as it gets depolarized, this one here can then open up, and the ones on sort of the trailing part of it are in an inactivated state. And so that basically, if you're at T0, this is what happens, and then all of a sudden the refractory period moves it forward. These ones become inactivated, and these ones here then go back to their closed. And in doing this, the wave moves down the axon. I hope that made sense to everybody. Think about it if you don't, if what I just explained, but it's just this refractory period where the channels are actually inactivated that prevents them from going backwards, right? These cannot open up, so it can't go backwards. Only these ones upstream can be opened when the membrane becomes depolarized, so it actually has directionality to it. Okay, I hope that made sense. You also have voltage-gated potassium channels. Here's just sort of a schematic of one. So normally it's in its closed state. When the membrane gets depolarized, it opens up. Potassium can then sort of flow out, and the membrane then gets repolarized, and it enters in an inactive state where it's sort of plugged, and then it'll go back to its normal resting closed state. All right, very similar to the sodium channels. Now, these aren't coded all over the place on an axon because the axons actually have insulation in the case called myelination, and the myelin sheaths that are here are actually made by glial cells to sort of protect the axon. And you can think of them almost as electrical insulation. You know how like on a wire on a cord. It's coated in sort of this rubbery coating, you know, that prevents you from getting a shock when you touch the wire. Same sort of concept here. And so what ends up happening is the depolarization occurs at these nodes of Ranvier, and they move in directions. Okay, so all of the voltage-gated sodium and potassium channels are going to be in these nodes here, as well as the sodium-potassium pumps. Here's a great EM of myelinated axon. Here's your axon here, and here's sort of the myelin that's going around it in the glial cell that's there as well. So this just simple wire insulation, if you want to break it down into its most sort of simple concept. Here's just a nice immunofluorescence of the nodes of Ranvier. Here they are, and here's their glia cells with the myelin sheet on it, right, to serve as sort of the insulation. And so the sodium channels are actually concentrated here in the nodes of Ranvier, and they cause the propagation down the axon. So a technique used in neuroscience is this technique called patch clamp, where you can sort of gently suck up a little portion of the membrane so that you have a single ion channel. And using this, you can sort of measure the current across it. Okay, and the patch clamp basically indicates, in studies using patch clamp, it's sort of an all or none. You can't have a partially open ion channel. It's either open and there's a free flow of ions, or it's closed. It's a simple enough concept. And here's just some real life patch clamp measurements. Here's the membrane potential, and here's your measuring the current in picoamps, and then when you get the sort of the aggregate current, you can see how the membrane becomes depolarized and then gets to be repolarized. Okay, so at synapses, and we've sort of looked at this before, but you have your sort of nerve terminal and your presynaptic cell, and then your action potential will be coming down the neuron, and that will then trigger the vesicles with the neurotransmitter to fuse with the plasma membrane, and we looked at how fusion occurs, and then the neurotransmitters will then go and bind to a ligand-gated ion channel, which then can open up the channel and cause depolarization of the next cell. All right, so this is how nerve cells connected will transmit signals. Okay, an action potential comes down, causes fusion of the vesicles, they'll release neurotransmitter, the neurotransmitters bind to a ligand-gated ion channel, and then you get depolarization of the target membrane. Here's a great EM. We've seen this before, I think, and you can see all, this is a nerve, a presynaptic nerve terminal, and here you can see the presynaptic membrane and the vesicles, and here is your dendrite or your postsynaptic membrane. Doesn't have to be a dendrite, could theoretically be something like a muscle cell or something. And here you can have another sort of area where you have all of your synaptic vesicles. Okay, and you've got, if you look down here on the schematic, you've got your presynaptic membrane, all of your synaptic vesicles, and then your postsynaptic membrane. And here in the postsynaptic membrane will be your ligand-gated ion channels. And so in the case with muscle cells, this is sort of a nice scanning EM of a neuromuscular junction. Here is the muscle cell, the myofibril here, and then you have your nerve termini, and here's your myelinated axon. And so ultimately what will happen is you'll get an action potential traveling down here, and ultimately the end result is the release of calcium from the sarcoplasmic reticulum, right? And we've seen that before. But how exactly does it work? Well, before we look at that, we actually have to look at the acetylcholine receptor. This is a ligand-gated ion channel, and so normally it's closed. When acetylcholine binds to it, it opens up. And then you can have it bound and sort of sort of closed. What's interesting is snake venom actually binds to this acetylcholine receptor. So if you sort of get bit by a snake and you become paralyzed, it's because the snake venom has bound here. Okay, and here's just the sort of the structural things of the acetylcholine receptor. Here's the acetylcholine binding site, and then you have your selectivity pore, and it opens up in the gate. So it turns out this is actually a pretty complex molecule because there's many different spliced isoforms and many different subunits, and these can all be mixed and matched depending on the cell and the cell type. So it'll have a different sort of response. And one of the things with the acetylcholine receptor, it's not selective for cations like the voltage-gated ion channels are that we saw earlier. So these, if they just open, things can freely flow in and out. Okay. So in the case with a muscle cell, here's how it all works, right? And we've already seen parts of this, and now we're going to see the other part of it. Okay, so here is your sort of resting neuromuscular junction. You have your acetylcholine in a sort of a presynaptic vesicle. And as an action potential travels down here, first thing it's going to do is open up this voltage-gated calcium channel. Okay, and when the voltage-gated calcium channel is opened up, calcium will flow in. The calcium and flowing in and the depolarization actually causes the vesicle to fuse with the plasma membrane, and acetylcholine is released. That binds to the acetylcholine gated cation channel. Sodium flows in. This causes depolarization of the membrane. Then you have opening up of the voltage-gated sodium channels, which then sodium flows in, and also it opens up a voltage-gated calcium channel. Okay, and then the calcium flows in, because once again the calcium is low in here. And then you have a calcium-gated calcium channel that we saw in the sarcoplasmic reticulum, and when calcium binds, a ton more calcium is going to flow out. And that's going to go interact with troponin and then allow a muscle contraction to proceed. So in general, at the neuromuscular junction, you have basically a sequential thing with 5 different ion channels. Right? You've got a calcium channel first, and then you have an acetylcholine-gated sodium channel, a voltage-gated sodium channel, and then a voltage-gated calcium channel, and a calcium-gated calcium channel. Okay? So here's basically all of the connections of a motor neuron in the spine, and you can see all of these sort of red spots are synapses. Okay, this is where the axon terminals are meeting up with the dendrites in the cell body. Okay, and you have this one important thing here called the initial segment. And the initial segment is sort of where the decision process comes in. Okay, this cell is receiving, tens of thousands, hundreds of thousands of different inputs, but not every input leads to an output where an action potential is going to go down the axon. And so it's this initial segment here that sort of makes the judgment call. Okay, so there's potassium channels and calcium channels, and they're sensitizing and desensitizing. And so what it's doing is integrating signals from all of these various inputs that it's receiving and integrates them to decide whether or not it's going to fire or not. And the way it works is you have this sort of post-synaptic potential. Okay, so here's your post-synaptic potential, and you're getting these signals, and that's causing the membrane potential to change. Not that big of a deal, but if it gets high enough, and there's enough of these, and it gets to the threshold, all of a sudden it's going to basically fire on down the nerve. Okay, and so you have these sort of inhibitory postsynaptic potential as well as ones that excite it, and depending on the ones that how excited it becomes, that will then sort of trigger and pass the threshold to send the stimulus, send the wave down the axon where you're getting depolarization and opening up of the channels in the nodes of Ranvier as you're traveling down. Okay, I hope that made sense to everybody. Okay, one of the last things we're going to look at is how this all works with learning and memory, or long-term potentiation. And so in this case, it uses the glutamate, and you have these two receptors, your NMDA and AMPA receptors. And so when you get an action potential traveling down the nerve cell, this will eventually lead to fusion of the presynaptic vesicles, and that will release glutamate, and glutamate will actually bind to the AMPA receptor, and that is a ligand-gated sodium channel. The membrane becomes depolarized, and then you have this magnesium plug in NMDA, which will then flow out, and that allows calcium to flow in, and it's this calcium that's flowing in that then leads to more sort of AMPA receptor inserted into the membrane. So the next time that glutamate is released, this will open up more and it gets depolarized more, and that is actually how we sort of remember things. So I hope you guys are getting a lot of this AMPA ligand-gated ion channel in all of your neurons as you're sitting here learning all of this stuff, because I really want you to sort of remember it all. Okay? And so just here's a little table of the various voltage-gated ion channel and neurotransmitter-gated channels. Serotonin, acetylcholine, glutamate, we looked at those. There's also GABA and glycine-gated, and here's some of your voltage-gated ones. Okay, and these can be either excitatory or inhibitory. So that's it. So next week, or next Thursday, we're going to do the extracellular matrix. So keep up on your reading. The extracellular matrix, some people will find it really cool. Others maybe not so much.